It's a pleasure to have Rob Tycko as our second speaker today. Rob obtained his PhD from University of California, Berkeley, working in Alex Pine's lab, and that's 1984. And then he went to Stanley Opela's lab at UPenn to conduct his postdoctoral work, developing, I think, N14 overtone NMR. In 1986, he joined AT&T Bell Labs, where he developed some of the earliest dipolar recoupling NMR techniques and studied some technologically relevant materials such as gallium arsenide quantum wells and C60. In 1994, he made a fairly big change of his scientific direction by moving to NIH. And his group has focused on a study of amyloid proteins, particularly the Alzheimer's beta amyloid peptide. Now, from studying disease-relevant proteins that misfold into multiple beta sheet structures over the course of years, it's not actually a far cry to wonder about how functional proteins fold into a single alpha helical structure very quickly in seconds. And because this kind of functional folding is fast, capturing the kinetics and intermediate structure is challenging. And I think that's what Rob is going to tell us today.
Thank you. Thank you very much, May. Can you hear me? I am going to talk about some unusual solid-state NMR experiments in which we're trying to develop and apply methods for studying transient intermediates in what I would call unidirectional structural conversion processes in biomolecular systems, and trying to do this on the millisecond timescale. This is a recent photo of members of my group after the COVID-19 shutdown. Most of the work I'll describe is work that's being done by my postdoc JQ and Jian. So I'll be showing the data I'll show is mostly from him. Wei-Ming Yao has contributed to this work by synthesizing peptides and synthesizing tri-radical compounds for dynamic nuclear polarization. Kent Thurber contributed to this work by constructing much of the apparatus that we use. Blake Wilson is currently working on related experiments, but I'm not going to talk about those right now. Okay, so there'll be three parts to the talk. I'm going to first spend 5 or 10 minutes just describing the concept of the experiments and the apparatus that we use. Then I'll show you some results for a particular system, the melatonin peptide, in experiments in which we can monitor the folding and tetramerization of melatonin following a rapid pH jump. That's published work. And then in the last part of the talk, I'll talk about unpublished studies that we're actually in the process of, we're about to submit that for publication, in which we're studying the process of formation of peptide calmodulin complexes in the presence of calcium following rapid mixing of the components of those complexes. And these are processes that occur, as you will see, on the 1 to 30 millisecond timescale in our experiments. So the basic concept is shown here. What we want to do is start with an initial state of a system that could be an unfolded state of a protein, an unbound ligand, or an unassembled peptide, and then do some rapid change in the conditions, the pH, the temperature, denaturant or mixing of two interacting components, and then follow the structural changes that occur after that rapid change in conditions. And then we can rapidly freeze trap the solution at various time points as that process occurs, and study the structural properties of intermediates, including the conformations and intermolecular interactions as that system proceeds to a final state that's the folded protein, a ligand receptor complex, or assembled peptide oligomer or fibril, for example. As you'll see, we can do these rapid mixing. We can change conditions on the millisecond timescale, the single millisecond timescale, and then we can rapidly freeze samples on the sub-millisecond timescale. So that sets the resolution of these experiments. As you'll see, we can go from about 1.5 milliseconds up to tens of milliseconds in these experiments. These are an important aspect of these experiments are that in most systems we're working with relatively low concentrations, less than 1 millimolar, and relatively small sample volumes, less than 100 microliters, perhaps 40 or 50 microliters in the actual samples. So we're talking about less than 100 nanomole of material. We're also doing direct carbon-13 or nitrogen-15 detection. So the sensitivity is an issue. As you'll see also, the NMR lines are quite broad. But these are experiments that actually require dynamic nuclear polarization to make them really feasible on a reasonable timescale. And so in my opinion, these are one of the more interesting things that you can actually do with low temperature DNP for biomolecular systems. So our DNP apparatus is a home-built system which is described in the literature. It's a home-built magic angle spinning probe in which we cool the sample with liquid helium, actually. So we cool the sample space within the magic angle spinning probe with very cold helium. We drive the magic angle spinning with nitrogen gas that actually usually enters the probe at room temperature. We use very long magic angle spinning rotors that separate the nitrogen gas from the helium. And although this sounds like maybe a preposterous idea, it actually works. And so we've demonstrated that in numerous papers. One important for our experiments, an important aspect of this system is that we can load a frozen magic angle spinning rotor, frozen in liquid nitrogen, into a pre-cooled DNP MAS probe and keep the samples at very low temperatures at all times. We just we can lower the probe out of the magnet with the helium transfer line attached to it, and then take one sample out, put another sample in just by hand in a matter of 5 or 10 seconds, and then start it spinning again. And that works fairly reliably. So there's no problem with the samples warming up, which is important because we're freeze trapping intermediate states. So we have to keep the samples at low temperatures at all times. So that's turns out an important feature of our experiments. We use helium to cool the samples. So we're using up helium. The helium consumption is less than 2 liters per hour for sample temperatures in the 25 to 30 Kelvin range. We can spin typically at 7 kilohertz magic angle spinning. The RF performance is sort of equivalent to the RF performance of a standard solid-state NMR probe. And with this system, we get, using an extended interaction oscillator as the microwave source, 1.5 watts maximum output. These are experiments at 9.4 Tesla, so it's 365 gigahertz microwaves. We get good, at these low temperatures, very large enhancements, up to a factor of 100, 150 sometimes, illustrated by this one example here. At very low temperatures, because the electron spin lattice relaxation is slow, you actually don't need very high microwave powers to get the full dynamic nuclear polarization enhancement by the cross effect mechanism. So you can actually use 0.5 watt, that would be sufficient. We can also do experiments at high temperatures, and we can still get reasonable enhancements. Most of our experiments are done in the 25 to 30 Kelvin range. We can get lower helium consumption by cooling the bearing gas, the nitrogen gas that we send in. We can reduce the helium consumption to about 1.5 liters, but we usually don't do that because that's just an additional headache, and the experiments are complicated enough as they are. So the bottom line is, with this equipment, we can, for the kind of samples I'm talking about, we can acquire good two-dimensional carbon-13 NMR spectra of frozen solutions in a matter of a few hours, 1 to 6 hours typically. If you don't do the essential to make this practical. I'm getting a message that my internet connection is unstable. I hope that's okay. So here's the apparatus that we use for doing the rapid mixing and rapid freezing for the experiments I'm describing. And this is, again, it's described in the literature, but basically we have a couple of HPLC type pumps that can pump solutions through a homemade mixer. And I'll show you the mixer in a second. The mixed solutions come out of the mixer as a jet. The mixer sweeps across the surface of a copper plate that is cooled to liquid nitrogen temperature. It's a rotating copper plate. The mixer sweeps across the surface. The jet deposits a film of frozen material on the surface of that rotating copper plate. With that kind of apparatus, again, as you'll see, we can freeze very rapidly. We can mix very rapidly. We can control the time between mixing and freezing. That's what we call the evolution time, by varying the distance between the mixer and the surface of that rapidly rotating copper plate, cold copper plate. So that allows us. We can also vary the pumping rate, the flow rates from the pumps. So that gives us a range of about 1 to 30 milliseconds. If we wanted to, we could go to longer time scales. with some modifications. The mixer was a key aspect of this. It's a home-built mixer just made out of simple HPLC fittings, basically a Y fitting, a Y adapter, where the inputs are the two solutions that we're mixing from the two pumps. In the exit from this Y mixer, the key thing is that we have a short section of tubing that's packed with stainless steel beads, 40 micron stainless steel beads. So as the solutions pass through that column, essentially, bead packed column, that's where they get mixed. And the time it takes to pass through that mixer itself at typical flow rates you use is about 1.6 milliseconds. We can pump a little bit faster. We can get it down to 1 millisecond roughly. And then what comes out of the mixer is a high-speed jet of solution, which, as I told you, strikes this rotating cold copper plate. The magic angle spinning rotor is loaded into a funnel-shaped piece in the center of the copper plate. So after depositing the frozen material, we actually lower the copper plate back into liquid nitrogen, and then we scrape the frozen material off the surface under liquid nitrogen at all times and pack it into the magic angle spinning rotor, keeping it at 77 Kelvin. While we're doing that, we can then take the rotor out and store it in liquid nitrogen until we're ready to do the experiments. And as I said, we can vary the time, this evolution time, by varying the distance, the flight distance, or varying the pumping rate. Just some evidence that the mixer actually works. To prove that, we can mix a solution of carbon-13 labeled glycine in glycerol water mixture. I'll just mention we use 20% glycerol with high freezing rates. That's sufficient glycerol content to get a good frozen glass. We mix a glycine solution with a solution that contains dysprosium, paramagnetic relaxation agent, and then we can measure, after freezing the sample, we can measure basically the proton T1 relaxation times of the solution. And these are data shown here. If we compare a rapidly mixed solution with a pre-mixed solution, we get the same proton T1s. These are carbon-13 detected by cross-polarization, but it's the proton T1 that we're actually measuring of the solvent. And so the fact that these T1 numbers agree tells us that the solutions are essentially completely mixed as they pass through our mixer. So that's one piece of evidence. Okay, now applications. So first I'm going to talk about two applications. The first is this melatonin peptide sequence shown here. Melatonin is a very widely studied peptide for a variety of reasons. At low pH, according to these CD measurements under our solvent conditions at low pH, the melatonin is a disordered monomer. As you raise the pH, as you neutralize it, it becomes helical at equilibrium. The structure of the helical form was solved by David Eisenberg's lab many years ago. It's a tetramer of helices with sort of a hydrophobic core. I'm not going to go into all those details. So it's a very widely studied system for a variety of reasons. It's a model system for protein folding, for pore formation in membranes. It's of interest as a cancer treatment even. It's used for a wide variety of purposes, even in biotech applications. So there's a lot of papers about melatonin, but there's still a lot of questions about how this process actually happens that are unresolved. But how does this happen as a function of time? Do the helices form first when you change the pH, if you do a rapid pH change, followed by the formation of dimers and tetramers, or do you get disordered tetramers first, and then you get an internal conversion from a disordered state to a helical state? What are the timescales for this process? So that's the kind of thing we want to be able to measure. For these experiments, we labeled just 3 residues, and you'll see that the spectra look very different from the spectra that Michael Sattler showed you. These are spectra with rather broad overlapping lines, particularly in the disordered state. Of course, you have a frozen ensemble, freeze-trapped ensemble of with a wide distribution of conformations, so you have big ranges of chemical shifts. There's no motions really. There's no motional narrowing or conformational exchange. So we see these very broad cross-peak signals in our 2D spectra, as you'll see in a second. Even in the fully folded state, there's always some disorder within the final complex. So even then, the lines are not very broad. We get line widths typically of about 2 parts per million, sometimes a little bit less, but not much. So we can't look at uniformly labeled systems. So the spectra that you'll see look very different, as I said, from the standard solution, multi-dimensional kind of solution NMR spectra. If you come at magnetic resonance from more of a material science, physics kind of background, or if you're used to doing EPR, electron paramagnetic resonance experiments, then you're used to seeing broad lines and it doesn't bother you that much. But it's different. It's a different type of NMR than some of you are used to.
Rob, are you still showing transparency 8, or have you advanced already? Because
it's still 8. It's still 8.
We still see 8. Okay.
Okay. I'm about to move on. Am I spending too much time on this transparency?
No, it was just we were thinking you were discussing Spectra already, and
I'm just warning you. I'm giving you a little bit of warning so you're not shocked. I don't want to shock you when I show the spectrum. Okay, just explaining a little of the philosophy behind these experiments. So the spectra are going to be kind of upsetting to some of you, but to me, I think they're okay. And as I hope you'll see, there's some information in these spectra, and that's the important thing. Okay, so here are one-dimensional spectra, double quantum filters, C13 NMR spectra, as a function of this evolution time I described by varying the distance between the mixer and the cold copper plate. And you can see that things happen on the time scale that we can actually probe. So here, the evolution time of 0 is just the low pH solution. Infinite evolution time is the neutral pH solution at equilibrium, and then everything else is what you get when you do the rapid mixing before freezing. And you can see there's kind of a progressive shift in the alpha carbon positions as helices form. There's a gradual narrowing of the NMR lines. That's what you see in 1D spectra. In 2D spectra, you start with these very, very broad cross peak patterns, even with this rather sparsely labeled sample. Eventually, you get to resolved cross peaks, but it takes some time for that to happen. So what we can conclude from these spectra just by inspection is that there's helical secondary structure that develops on the 5 to 10 millisecond timescale roughly, but full structural order develops later because the signals continue to become narrower, the cross peaks sharpen up even at much longer times after you have helices. You can also see that isoleucine 20 is already partially helical even at the very beginning. One way of analyzing these spectra is by doing a principal component analysis. You can mathematically derive a set of basis spectra that exactly describe the experimental spectra, and then you can look at what how the coefficients of these different basis spectra vary as with the evolution time. One thing that comes out of this is that there are actually three. We can demonstrate that there are actually three principal components that are statistically significant, that are not just due to noise in the spectra. So we believe that the melatonin tetramer formation is not a simple two-state process. And from these, you can sort of, from the buildup of, say, the second principal component, the time dependence of that, you can see that things are happening roughly on the 2 to 5 millisecond timescale, but then things continue to happen later, and that's a gradual annealing of the structure, we believe. We can also measure, because of the way our labels are positioned, we can measure intermolecular contacts, which are attributed to the tetramerization of the peptide. We can look at buildup of cross peaks between isoleucine 20 and leucine 6, which become close together in space in the final tetramer, but in the monomer are not close together. We can also look at glycine 3 to leucine 6, which are intramolecular cross peaks. And you see the intramolecular cross peak and the intermolecular cross peak build up again on more or less the same time scale. So it seems to be a concerted process. So you don't get a rapid helix formation followed by tetramer assembly. The whole thing seems to happen more or less at once. And there's more details of this that you can read if you're interested in this, details of the experimental apparatus and what we've learned about melatonin in this paper that was published about a year ago. This is really our first paper describing these kinds of experiments. I want to talk now about unpublished results that we're about to submit for publication, where we're studying another system that's of very wide interest. That's the calmodulin and its complexes with various target peptides and target proteins in the presence of calcium. Of course, it's a very widely studied system in the NMR community, of course, by solution NMR techniques. It's biologically one of the extremely important protein. It's extremely highly conserved all the way from fish to mammals and humans. There's no changes in the amino acid sequence over hundreds of millions of years of evolution, and actually humans have 3 calmodulin genes, all of which produce exactly the same amino acid sequence. So it's a very unusual and very important protein because it's the main mediator of calcium responses within cells, and it really affects everything that cells do. The system that we're studying, so what's known is that calmodulin in the absence of calcium has a structure that's been studied by solution NMR with a lot of flexibility, certain helical domains. When it binds calcium, the structure undergoes a major conformational change. The helix bundles, there's 2 domains, each of which forms a 4-helix bundle. And that creates a hydrophobic cavity or tunnel or channel into which a target peptide can insert. And typically the target peptides, which are the calmodulin binding domains of calmodulin activated proteins, they transition from an unfolded state to a helical state when they form this complex. So how does this actually happen? There's major structural changes in both the target peptide and in the calmodulin itself. How does this happen as a function of time? Does it all happen at once, or are there different stages that we can identify? So to study that, we're rapidly mixing a calmodulin solution with a solution of M13. That's the peptide shown here. This is the so the calmodulin binding domain of skeletal muscle myosin light chain kinase, which was one of the important systems that was studied originally by Ikura and Bax and my other colleagues, Angela Gronenborn and Marius Klor at the NIH. It's a widely studied system. That's what we're studying. We can synthesize the M13 peptide. We can put isotopic labels into M13. We can also, in the experiments I'll describe, we also put C13 labels just at the methyl sites of methionines, because contacts between methionine side chains and target peptides are important in the final complex, so we can detect intermolecular contacts as well. So here are spectra. Again, broad lines, just the M13, a solution of M13 with calcium by itself, extremely broad lines, although there are only, in this case, 4 amino acids that are isotopically labeled. This is the fully formed complex. If we just mix the 2 components and then freeze them, this is what we get. So the 4 labeled, in that state, you can resolve the C alpha, C beta cross peaks of the 4 labeled sites in this case, and you can see that the chemical shifts indicate helical structure, and you can see that from connecting those to the unbound state, you can see that there's really not much helix content in the unbound state. And then we can look at spectra as a function of the evolution time, and you can see there's a gradual change in the spectra. After the shortest time that we can sample, 1.5 milliseconds, there's clearly a mixture of broad components and sharp components. And at longer times, the sharp components predominate. So then gradually there's a reduction in the population of conformationally disordered M13 and an increase in the population of alpha helical M13. And we can, from looking at different spectra where we subtract the fully folded, the fully formed complex signals from the signals as a function of time, and then look at the remaining cross-peak volumes, which we attribute to the disordered components. We can look at buildup curves of the structurally ordered components on a residue-specific level. And the important thing is that we've looked at 2 different labeling patterns. 3 of the labeled sites, 4 of the labeled sites, which are towards the N terminus of the peptide, become fully helical within about 2 milliseconds, whereas sites that are near the C terminus, residues 17 and 20, become fully helical later. So this is the first indication that there's actually 2 different timescales. We can also look at intermolecular cross peaks because we label all the methyl carbons on methionine side chains of calmodulin, and there are close contacts of around 5 angstroms or less to the sites that we've chosen to label in the fully formed structure. So we can look at these intermolecular cross peaks and look at the buildup curves of intermolecular cross peaks. And again, we see towards the N-terminal end a rapid buildup of intermolecular contacts, again on about 2 millisecond time scale, and a slower buildup of intermolecular contacts towards the C-terminus of the peptide, again on like an 8 millisecond time scale. So it seems that what our data are saying is that the N-terminal portion of M13, the calmodulin binding domain of the skeletal muscle light chain kinase, The N-terminal portion converts from a conformationally disordered state to an ordered alpha helix in about 2 milliseconds. The C-terminal portion becomes alpha helical more slowly. Now, the N-terminal portion interacts with the C-terminal domain of calmodulin. And so phenylalanine 8, which builds up its intermolecular contact quickly, has a short contact to methionine 145 in the C-terminal domain of calmodulin. So that contact builds up quickly, whereas the contacts to the N-terminal domain of calmodulin, to methionine 51, methionine 71, and methionine 72, those develop more slowly. So what we're seeing, this is the finally formed structure in the in the structure that was deposited in the PD by Grishayev and Clore and Anthus. I think this was 2012, maybe. So this shows, here I've colored the C-terminal domain of calmodulin interacts with the N-terminal domain of the peptide. The N-terminal domain of calmodulin interacts with the C-terminal domain of the peptide. And we're seeing this part of the structure developing in about 2 milliseconds. This part of the structure develops at about 8 milliseconds. Even after that, even after 8 milliseconds, there's a still continual narrowing of the solid-state NMR line. So the level of structural order continues to increase with time, even after you get a structure that looks approximately like this. So the bottom line is calmodulin is a crucially important and heavily studied protein, but so there's many papers using other techniques, stop flow fluorescence techniques, time resolved small angle X-ray scattering techniques, and other techniques where they've tried to study this kind of thing. But this level of detail really for the molecular mechanism of complex formation has not been achieved by other experimental methods. So I think this is a real contribution. It's easy to imagine many other applications of this kind of experiment. And so we're planning to pursue some of these applications in the near future. Thanks very much for your attention. If you have any questions, I'll be happy to try to address them.
Thank you, Rob. So there are 2 questions in the Q&A. I'll start with these. And so the first question from anonymous attendee is, what other systems has your freezing apparatus been applied to? For example, IDPs, membrane proteins, where there are any issues seen in changes to freezing compared to a liquid sample seen done in solution NMR? And oops, this jumps. And was, oops, this is jumping around. Was glycerol applied to the samples, and did the presence of glycerol appear to change the behavior of the proteins? So several questions.
Okay, so I guess the bottom line is this okay? Sounds like the question is basically, the information we get from these experiments, is that information really relevant to what happens at physiological temperatures or at room temperature? I think that's the gist of the question, right? So there is glycerol added so that we get a glass. We're using 20% glycerol, relatively low. We've seen no evidence that the freezing process, and we've been doing experiments of this sort for many years actually in the lab, looking at frozen solutions, not with this kind of time resolution. It's a common concern, and I guess in other communities, the EPR community, I think this was a concern that was raised with respect to many EPR experiments that were done on frozen solutions for many years. And I mean, I think historically, once crystallographers started cooling their crystals after adding cryoprotectants to their crystals, and then they found that that was okay. So that crystallography is always done at low temperatures, and of course now cryo-EM is because it's cryo-EM is done at low temperatures. I think some of those concerns are less prominent compared to what they once were. But I mean, you have to be aware of that, and it could be that in the time scale, if it takes us let's say half a millisecond to freeze our sample, which is probably an upper limit, maybe it's less than that. I mean, something could happen on that. If there's a strongly temperature-dependent stability of some complex, maybe it starts to unfold or something as we're freezing it. There's some proteins undergo cold denaturation. I mean, we've never seen any evidence that that's actually happening. But you can imagine that that's happening. One thing to keep in mind is these are glycerol water solutions. They become increasingly viscous as you cool them down as well. So you don't have to, the relevant time is not really the time from room temperature down to 77 K. It's really from room temperature down to 0 C. once you get below 0C, the viscosity of the solution is very high, so any conformational changes are going to happen even more slowly. So I think it's really pretty safe. As far as other applications, I mean, there's other things. Protein folding, we're working on just straight, globular, monomeric globular protein folding with actually using temperature jump experiments that we're working on. Applications to initial stages of amyloid fibril formation is something that we're probably going to look at. I think insertion of peptides that say antimicrobial peptides that insert into biological membranes, the initial stages of that process, you could imagine mixing vesicles or bicels or something with a membrane interacting peptide, and then following the time dependence of that membrane insertion or pore formation or channel formation process. That would be an interesting thing to do. I'm now thinking, is it maybe there's something we could even do with cells, where we take cells and probably not mixing them quite this quickly. That would probably disrupt the cells, but do some kind of transient effects on cellular responses to something or other. I don't know. We're just.
So maybe I can add.
I welcome any suggestions. Go ahead.
Maybe I can add to that question a little bit by asking, whether you see a correlation between your low temperature M13 insertion result with high temperature dynamics measurement by solution NMR, maybe between the N-terminus and C-terminus. Maybe there's a connection there. The fast folding.
Well, I think it's very different. What we're doing is very different. So I don't know if there should be a connection there. I mean, obviously this I should have, but it's it's a good point to raise. I mean, obviously there's a lot of studies of dynamics, and we've heard one example of that from Michael Sadler. The difference here is, usually what people study by conventional NMR techniques is the dynamics that are occurring at thermal equilibrium. What we're looking at is a very non-equilibrium process. We're looking at a approach to equilibrium from a situation that's initially very far from equilibrium, created by our rapid mixing. So it's qualitatively different. I think the comparison that you could make is to optical measurements, really stop flow type fluorescence techniques. And what we're seeing for the calmodulin system is related. There's an important point that stop flow measurements are usually done at very low temperatures. I'm sorry, done at very low concentrations. At very low concentrations, the kinetics that you see are usually dominated by just the initial encounter between the interacting components. The time it takes for a ligand to make a productive collision with its receptor. That's what you actually measure. Well, under our conditions, we have much higher concentrations. So the initial encounter happens quickly, and then we're studying the process of structure formation within that complex. And that's been hard to get, that people really can't get at that very well from stop flow fluorescence. So a lot of systems that look like just simple two-state systems by stop flow fluorescence, they look simple because the experiments are done at very low concentrations. And under our conditions, so they don't actually see the internal structural conversions.
Yeah. Okay, so then one can get into a discussion of the concentration in more in vivo setting. But I think I'll move on to some of the other questions. So Daniela Goldfarb has a question. Does it matter if the calcium is added to the protein or to the peptide?
Yeah, in our experiments, that's a good question, and we should do those experiments. We probably will. In our experiments, we're mixing M13 plus calcium with just the apo calmodulin. So it would be also interesting to first load the calmodulin with calcium and then mix it with the peptide and see if the process is somewhat different. We haven't done those experiments yet.
Mm-hmm. So these questions jump around, but I spot an interesting question about alpha helical beta sheet propensity of the Militing story. I don't know why it's jumping around. So can you comment on the helical or sheet propensity of unfolded Militing or M13? Is it possible to derive any information before the protein folds, such as the distribution center that coil chemical shifts at the speed?
Well, you can look at, you can look at this, I guess the line, these very broad crossbeak line shapes that, to just go back, just to, so if you look at this, you still see my cursor. So, here we're basically seeing the conformational distribution of M13. So you can try to interpret that. We've done experiments along those lines in the past, trying to separate out different conformational components actually to get a conformational distribution. And you can also, we've done experiments on the HP35 protein in frozen solutions as a function of denaturing concentration. and try to extract the conformational distributions from actually doing quantitative measurements of torsion angles and intramolecular distances, that kind of thing. That was a long time ago. But you can actually try to quantify conformational distributions from these kinds of measurements in the frozen solutions. Those kinds of things can be done, so you could try to tease out the beta sheet propensity of the peptide by itself. We haven't done that in this case, but you can see from these just comparison of these two spectra that there seems to be very little alpha helical propensity. In the case of the melatonin, one of the labeled sites, isoleucine 20, even in the sort of the low pH state, seemed to have intensity at the alpha helical position. So it was partial population of the alpha helices.
Mm-hmm. I'll ask another question from water chasing, and then maybe we can go to the informal session. So very nice result for calcium, calmodulin and M13. I may have missed this, but did you add calcium, similar to Daniela's question, or was calcium also added when mixing? Have you looked for a correlation of the calmodulin interaction with them?
Yeah, it's the same sort of the same question. In this case, we are taking the M13 calcium solution and mixing that with the calmodulin solution. And the calmodulin solution is prepared in such a way that it should not have any calcium already bound.
Mm-hmm.
But I think looking at it the other direction, I mean, in a way that I guess that's, in a way, I don't know what you mean. That's arguably the more biologically relevant thing is, you already have the assembly process is triggered by the influx of calcium into the cell. So starting with calcium that doesn't have, or starting with calmodulin that doesn't have calcium bound is maybe the more biologically relevant, but I think both conditions are interesting. So we should do both.
Rob, can you hear me?
I can hear somebody.
Yeah, it's Walter Chazen.
Hi.
I just wanted to add that, so the second part of this is that there's a very interesting correlation that you see with the binding of calcium to calmodulin.
Right. Yeah, there's a little, so it's believed that that's a good point. I didn't go into all the literature, but the C-terminal binding sites, the C-terminal EF hands are supposed to have higher affinity. So the affinity sort of, or the order, there's sort of an order of binding of calcium that's been identified, which proceeds from the C terminus to the N terminus.
Yeah.
By previous measurements.
Go ahead.
And there've been some fancy stop flow and continuous flow fluorescence experiments where they just look at what happens when you mix calmodulin with calcium, and there's evidence that kinetically these sites get populated first, and then these get populated later. Actually, the timescales they see are even slower than what we see. So maybe the kinetics of calcium binding to the N-terminal domain is accelerated when you actually have a peptide that's partially bound.
Yeah, energetic coupling of the two binding events.
Right.
Beautiful.
Right. But I mean, also kinetic. I mean, it has this kinetic effects. It's not just the affinities.
Yeah. No, absolutely.
Okay, great. So since it's 20 past 11, the official session, we'll call it over now, but people are welcome to hang on for more questions. I know there are more questions for both speakers. So thank you, and we'll see you next Thursday.
Thank you very much. Thanks again for inviting me.